In this video, we will learn about competent cells. Competence is the ability of cells to take up externally present DNA. Cell competence has become an essential research tool in the field of genetic engineering. It provides us with a mechanism to introduce foreign genetic material into a host cell. The competence can be naturally present. This has facilitated a wide range of adaptations in prokaryotic and eukaryotic cells. Some of the known examples include Vibrio cholera, the causative agent of cholera. These bacteria lyse neighboring cells to obtain nutrients, but also scavenges the DNA released from their victims. The capacity to take up foreign DNA can be artificially induced by specific treatment of the host cells. During this video, we will learn how to prepare and evaluate competent cells. The model organism we often use in molecular biotechnology, Escherichia coli, is not naturally competent. Therefore, to use these cells for cloning or expression studies, we must induce competence. There are two main types of competent cells: electrocompetent and chemocompetent. Electroporation, or chemical transformation, creates temporary pores in the cell's membrane. These pores can be used to intake foreign DNA. The choice between the two methods is determined by several factors. These include the transformation efficiency that is needed for the experiment, the size and quantity of the DNA to be transformed, and the available equipment. Chemical transformation can be performed in a simple lab setup, yielding transformation efficiencies sufficient for routine cloning. Electroporation tends to be more efficient. That is why it is the preferred option for applications such as library creation. During this course, we will focus on the preparation and evaluation of electrocompetent cells. Let's see briefly how the electroporation leads to the intake of foreign DNA. When voltage is applied, the electrical pulse rearranges the orientation of the phospholipid bilayer. This happens in a way that leads to the formation of temporary pores. These pores then allow the intake of foreign molecular material, such as DNA, into the cell. By applying the appropriate voltage during the electroporation, the membrane permeability of the cells increases. However, cells can also survive the effects of membrane permeation and recover. Now, we will focus on the standard workflow for the preparation of electrocompetent cells. The first step is to streak out the desired E. coli strain on an agar plate and let the cells grow until colonies appear. Then, we prepare the overnight culture. This is done by inoculating a single colony from the fresh agar plate. The overnight culture is grown at 37 degrees Celsius with agitation. The next day, it is used to inoculate the main culture. The main culture is grown also at 37 degrees Celsius with agitation until it reaches an optical density of around 0.6 to 0.8. Further on, the cells are harvested via centrifugation. From this step, we continue working on ice. The cell pellet is resuspended and washed several times using ice-cold water or 10% glycerol. Finally, the cells are stored in 10% glycerol at -80 degrees Celsius until further use. The cells can be used for several months, but remember that upon prolonged storage, the competence decreases. It is important to highlight why we harvest cells at a specific optical density. As mentioned before, in a standard protocol, this is at around 0.6 to 0.8. Here you can see a scheme of a typical microbial growth curve. The growth curve is composed of 4 phases. These include lag phase, log or exponential phase, stationary phase, and decline toward the end. During the log phase of the growth, the cell mass undergoes several cell doublings. At this phase, the specific growth rate of the culture remains constant. This phase is also called the phase of balanced growth. This is because cells are as close to being metabolically identical throughout this period as they can be. That helps us to minimize experimental variations and have unified transformation efficiency across the culture. Once we have finished the preparation of competent cells, it is important to test their transformation efficiency. As mentioned, transformation efficiency is a key consideration when choosing applications for competent cells. Here, we will see a typical workflow for the transformation. This workflow is used to evaluate transformation efficiency. It is also used to routinely introduce a newly cloned plasmid, for example, coming from a Gibson assembly. The first step is to thaw electrocompetent cells on ice. Then, we mix salt-free plasmid DNA with the cells. Further on, the cells and DNA are transferred to an electroporation cuvette kept on ice. This cuvette has 2 aluminum electrodes on its sides. The cuvette is inserted into the electroporator. An electrical pulse is applied across the solution using electrodes. The success of the electroporation depends greatly on the purity of the plasmid solution, especially on its salt content. Solutions with high salt concentrations might cause an electrical discharge. After the electroporation comes the recovery phase. This is when rich growth media is added to the cells and the culture is regenerated. This happens for an hour at 37 degrees Celsius with agitation. After the recovery phase, an aliquot of the cell culture is plated on the respective selection plates. Finally, to evaluate the competent cells, we need to calculate the transformation efficiency. For a test transformation rate, we use a plasmid sample of a known concentration. The efficiency of transformation is typically measured as the number of transformed cells per microgram of DNA added to the cells. Several factors affect transformation efficiency. To name a few are plasmid size, forms of DNA, the genotype of host cells, and the method of transformation. In practice, the rates are higher for smaller plasmids and considerably lower for larger plasmids. Here, you can see some examples of good transformation efficiency for chemocompetent and electrocompetent cells.